Hey, good afternoon. You all have requested for the pre-recorded session of this, so I thought we'll do it today. So this is all about how to publish a first-time barcode manuscript or a research article. Okay, manuscript is a previous form of research article before peer reviewing and publishing. So since we have wide range of audience, we will see how to start from the scratch and take it to how to preparing the manuscript. All right. So research publication, what is that? We have school students, college students. You might have read a lot of textbooks so far. Where does this information comes to textbooks? They're all coming from research articles. Once scientists find something, they want to communicate for public social well-being. They will write their findings as a research manuscripts, and peer reviewers, editors will review them. If there is a necessary changes, they will make it, or else if it is very useful for society, they will publish it immediately. That's how you have a textbooks. All textbooks, the data comes from research article. That's why it's very important when you do a little research, whatever is your research finding, it's very important to communicate. You will see all your progress. If you are a BSc students, if you are aspiring for a European country MSc, or if you are a MSc aspiring for PhD, you need to have a research article because that's a proof of your skill. Your college degree or your workshop certificates will not act as a proof. The research articles are the proof of your skills. So these are the types of research articles we have: journal publication, book chapters, conference proceedings. What we are going to see today is about journal publication. Why? Because we intend to publish our manuscript as a all first time barcodes. We had a plan of publishing. Those who have taken species that is already barcoded, you also have an option of defining a barcode gap for the species or genera so that it can be a new data. See, having a new data is very important for publication. Any reviewer or editor, they are looking for new data. Those who got rejected multiple times from various journals will know this. The word novelty, lack of novelty will be there on all your rejection letters from the editors. Okay. So having a new data is very much important. So this session is all about how to prepare manuscript with first time barcoded species. So journals, every journal have their own style, formatting, number of words and everything. So you cannot simply prepare one manuscript and communicate to all journals. Okay, you need to have it them as a template for every time when you communicate to a journal, the manuscript should be optimized. Okay, so what are the steps necessary for communicating a manuscript after preparation? Probably these are the things you need to find. You need to identify a suitable journal. Okay. Elsewhere and Springerlink have something called Journal Finder. You can just copy your abstract, paste it there. They will suggest you the journals. And then you need to optimize your manuscript for the journal. And while submitting cover letters, whether it's through submission platforms, you need to follow it. Then it will be subjected to peer review process. After peer review, the revisions, if required, will be communicated to the authors, in this case, you guys. And then if it is accepted, it will be published online first, and then the hard copies will come out. So after publication, how many citation your article receives will decide how impactful that is for the society. Okay, so these are the 5 or 7 steps. So today's purpose is just to understand how to prepare a manuscript in first time barcoded species and how to define a barcode gap. Okay, for MSc and BScs, please take a look here. This is one of my message from a foreign university to my student, clearly describing what are the conditions they had and how the student did not meet those conditions. That's why their application is rejected. Okay, so these are the messages you will naturally get if you don't have a publication along with your degree before you apply for anything. So let me jump directly into how to prepare the manuscript. These are the way. A manuscript structure should have all these 5 important components: abstract, introduction, materials method, results, discussion, reference. Why did I name them? Why did I number them differently? This is the order I use to prepare manuscripts. Okay. I usually first write my materials method, then the results. I will describe them, then the discussion. For discussion, I need to read a lot of many manuscript to compare my data with their data. So once you have read many manuscripts, then you naturally qualify to write the introduction because you have seen how the introduction are read are actually written in all those manuscripts. Then finally references. When everything is done, write an abstract, go for journal finder and find your suitable journal. Okay, so journals have paid journals. You need to pay if you want to publish it. I strongly recommend not to go for that. There are actually closed access journal where readers have to pay and read it. In that case, authors don't need to pay. So hardly I pay for my communications. Okay, so today I will show you how I publish a manuscript. Okay, so this is a manuscript. This is one of my publications. So as I have told you many times, I'm not a zoologist. I'm just a molecular biologist. I specialize myself in DNA barcoding, but any phyla they comes to me, plant or animal, I know how to barcode it. So in this barcoding paper, we took 15 days for sampling and 10 days for writing, and we waited 30 days for publishing. That's it. In 45 days to 50 days, we got the publication online. So how did we do that? We sampled 4 species of isopods. Okay. When I barcoded them and subjected to blast analysis, I discovered Bathynomus kinsley and Bathynomus decumspinosus did not have a barcode. They are first. They have been barcoded for first time. How do I know that? Like I shown you in the previous video, see the variations. It is more than 7% variation, meaning that sequence is definitely not there in the database. And other things are also there. They both shared more than 7% variation. We know that another 2 species had less than 3% variation, meaning more than 97% similarity in the blast analysis. That's how I know they are not barcoded for first time. So I just mentioned that inside the manuscript, but in the title, I just highlighted only those 2 species that is been barcoded for first time. This is point number one. So how to write an introduction? All introduction will have what is been existing, what was the problem. Say here, what was the problem associated with the giant isopod taxonomy? How did we solve it using DNA barcoding? Okay, how can that be solved? We have proposed this. Materials method should be very detailed. When did I collect the sample? How did I collect the sample? What is the DNA extraction procedure used? PCR, what kind of primers are used? Okay. If you have outsourced a sample for sequencing, you clearly mentioned that I have collected these. These are the key morphological features of my sample, but I have outsourced the sample and the DNA isolation. This primer has been used and this machine has been used for sequencing. You should not say that I ran the gel and everything, which is ethically not right. When you outsource a sample, clearly mentioned that the sample has been outsourced and the results has been obtained from the commercial sequencing. But I bet you guys understand this entire paragraph, which is I think you have skills in these aspects now. So the machine output file received from the company has been subjected using, ChromaSlide, and they are compiled. You have taken for blast analysis, after which they are, they are checked using bioedit, and ClusterLex has been used for multiple sequence alignment, and Mega has been used for phylogenetic construction. Okay, and finally K2B distance analysis was done, which we will also cover in this session. How to calculate barcode gap. Okay, so I guess Metals method is very clear. You can write Metals method for your sample. Those who have taken the species yet to be barcoded species are those who have right now successfully barcoded the species for first time. I think many of you have received your sequencing results by today. I think from morning we are sending and only very few are left. Most of you might have received by now. So you need to document the key morphological features. For example, some are working with peace lily, a particular species of that. For declaring that specimen to be that species, you should know what are the key morphological features that species possess. You should clearly describe them in your materials method itself. Okay, so I wish you guys should start writing the introduction of your manuscript. If you have yet to be barcoded species, focus on the morphological features. If it is already barcoded species, focus on the barcode gap. So what is this barcode gap? We will see. So result section should be following the same order you have mentioned in the introduction, the specimen collection, and so we are mentioning about the accession number. Okay, those who have done the sequencing, you should know what is accession number. You are supposed to submit your sequences to GenBank and you get the accession number, because the reviewers and editors will check when you declare them as a first time barcoded. Have you really barcoded? They will also check in the NCBI. So how to check this accession number? Um, you please read your email. At the bottom of the email, they have asked you for 6 questions. Answer the question and reply the email immediately, at least today, at least. More you wait for a longer time, this GenBank submission process will be delayed. Okay. So once you submit the sequence to GenBank, NCBI GenBank, they will give you an accession number. So just copy that accession number, go to GenBank. I will show you how to check an accession number. Say, for example, I have a GenBank here. I will paste my accession number. I selected nucleotide. Okay. I have pasted my accession number. Search. So that's it. This is my sequence page. The DNA data that has been published in 9th March 2020. And here is my name, affiliation of the institution that done the work. Okay, all those data are there. This is very much important. You should get it as early as possible. Okay, these accession numbers will be cross-checked with the editors and reviewers before they find it as really it is first-time barcoded. So I am talking about blast analysis results here. So today I will share this paper also in the group. Use it as a template if you're working on a plant, or find some if you're working on an animal. Sorry, find some similar plant templates which can you use to describe your results for manuscript preparation. See, another species has only 93% with some other species. That's how I know that it is first time barcoded. The editors will also check using your accession number. So the phylogeny I will describe. I prepare a blast table where I gave this is just DNA barcoding giant isopods, that is DBGI. I gave some numbers, and here is the morphological identified species and their blast similarity. These are the accession number of the blast similar species, and my accession number has been given in the. So where did they actually isolate it? When you read the accession number of your reference sequences, you will know whether that sequence has been published in a manuscript, or if so, who is the author. Everything has been there. You can compare it. What I have done here is I have drawn the phylogram in the, actually I think it's in PPT PowerPoint. I just pasted the phylogram. I pasted my specimen images, cladding to the matching to the phylogram branches, and then I took it as one more picture and I have communicated it. Okay, that's how I compiled this picture. It looks nice if you incorporate your morphological features in the phylogeny. Okay. So what is this pairwise distance data? I'm very sure and pride that you guys by now I have seen many Excel sheets. You have done a very good job. See, if I want to calculate a pairwise distance data for Diodorlini, you see, I know I have only 2 sequences of Diodorlini, right? I have only 2 sequences here, 1 and 2. So 1 and 2. This diagonal 0 means, say for example, this 0 represents the distance between third species and third, which are of, when you compare the distance between the same DNA sequence, it's always 0, right? So, but this is missing in the data, in the Excel sheet. You should know that this is just an empty blank. When you calculate it in the mega, you have to introduce this 0 along the diagonal, because whenever they are meeting, when they are intersecting, that the value is 0. Okay. So for Diodorlini, if I want to calculate, I have only 2 sequences in the NCBI. I have only one sequence, and one sequence is produced by me in this work. So if I want to calculate barcode gap within Diodorlini, I will include only these 3 data. 1, 2, 3. Because Diodorlini belongs to only these 3 cells, right? 1 and 2, 1 and 2. Other species are here. Okay. So 0 plus 0.01 plus 0. These 3 values divided by sum of all these 3 divided by 3. That will give you the average barcode gap within Diodorlini. Okay. If I want to calculate barcode gap within Giganticus, so Giganticus, I have only 3 sequences. Only 2 was there in the GenBank, and 1 sequence is mine, 7, 8, and 9. So if I compare 7, 8, and 9, these are the sequences, right? So 1, 3 plus 2, 5 plus 6. All sum of all 6 values divided by 6 will give you the average barcode gap of Giganticus. That's how you calculate the barcode gaps within the species and also between the species. If so, I know the average barcode gap, say for example, for Giganticus is 0.01. If I want to compare it with Giganticus and Jas Messi is only 1 sequences, okay, which is 6. I can simply compare it with the 6th value, where Giganticus and Messi actually producing the data. Okay, so you will know the barcode gap between the species also. That's how the barcode gaps are calculated actually, which is very much important. You see, not only I have barcode, barcode of these species for first time, I have also proposed a barcode gap. We recommend a minimum variation of 3% variation based on genetic distances as a barcode gap to differentiate. Why? Only when you propose a barcode gap and someone comes with unknown sequences, when they do the blast, they can make a call whether it has been already barcoded or not. When it is falling within the cutoff range, it is barcoded. If not, it is not. Okay. So, or identify species in Bacillus genera, or until a new barcode gap. Why I say new barcode gap has been suggested after sequencing most species? Because for most species, I have generated two first-time barcodes, and other species don't even have a barcodes. And other have only one or two barcodes. I don't know. Maybe in near future from Australia or from other countries, many such species are barcoded. Then we will know that the barcode gap has to be increased or decreased. Okay. So it is very much important that we have this option, an open-minded option, that this is the barcode gap I'm defining. If more species are sequenced, especially in case there are no more reference sequences in the database, we have to have an open-ended question that more barcode gap can be included. Actually, this suggestion is given by my reviewer, not me. So I have included it, and I think this journal is around 2.3 impact or something. It got published. Okay. So discussion, you will compare your data with other data. You have to justify why you call them as a particular species. That's why it's very important for you to carefully document these species characteristics. Okay, for isopods, the spines, the I don't know, the carapace, morphology, many things will play a role in identifying the isopods. So it should be very much careful in describing them, documenting them. Then only we can complement them with the DNA data and go for a publication. Okay. References, this is an API style. Every journal have their own styles. So that's why you can have a template, but you need to optimize it for every time you communicate to a particular journal. Okay. So I wish after today, I want to see you immediately replying to the emails and get the sequence published, get the GenBank. The NCPI will email you directly to you with the GenBank accession number. Second thing is prepare your manuscript as early as possible and communicate it so that we can start communication process. Mostly maximum of 3 months can be expected before acceptance. So do not delay. Let me see your manuscript in the email as early as possible. Thank you, guys.